I'm excited to be here to be able to give this talk. I am at the University of Virginia, and I hope, I'm wearing my mask just to be safe, but I hope everyone can hear me well. I should say this is an exciting place to be, not only talking about basic science research, which is my world, but also to interact with those of us who are caregivers and family members. And I want to preface it saying that I am not usually in that space. And so beforehand, if there's something I say or somewhere I say that's insensitive to families, please forgive me ahead of time. I'll try to be conscious of that. But again, I work with mice and I study mice, but we're excited about some of the work that we're doing. So this is my lab. We're located in the University of Virginia. We have a diverse group of researchers. We study the microglia, which I'll tell you a little bit about, some of the work that we do. We have postdocs, grad students, technicians, and we're excited, really excited about these microglial cells. We're in the Brain Immunology and Glial Center, as well as in the Department of Neuroscience, funded by several sources. And then also, I am appreciative for my family who gives me the opportunity to do this work, as well as to be here today and be away from them. So in terms of the outline of my talk, mine will be slightly different. We've heard a lot of talk about the SCN1A. We're starting to get into the Dravet syndrome world, but our work is actually focusing on these cells in the brain called microglia, and I'll tell you a little bit about them, and then lead into the main story of our work broadly in seizure disorders before we end with some initial work that we're starting to do in Dravet syndrome. And I'll summarize that and show you our future direction. So to start off, we're interested in these cells called microglia. For those of us who don't know, the brain is made up of neurons, as everyone knows. But the brain is not only made up of neurons, it's made up of these other cells that are broadly called glia. Glia comes from the Greek, which meant glue. And that's what we thought these cells did. They were simply structural elements of the brain that helped to keep the neurons intact. We now know that that's not the case. They actually play very important roles. And in these schematics shown here, the neurons are depicted in this case in brown, and in this case in cyan. And what you can see are these glial cells, several of them. For example, this green one that is called the astrocyte, or shown here in green as well. It's interacting on the one hand with the neuron. It's interacting on the one hand with the neuron, but also with the vasculature. And these cells are considered to be the kind of the stars of the brain because they have these processes by which they can interact with different elements. And they regulate the vasculature as well as regulate neuronal function. There are these other cells that are depicted here in magenta, and they wrap processes around neurons to facilitate the electrical conduction of impulses in the brain. So for individuals with problems with the oligodendrocytes, you actually have a problem in the sending of signals and electrical impulses throughout the brain. But also, while these are exciting cells called known as glia, I do not study any of these cells. I study a different type of glia. Actually, I think it's the coolest type of glia. That's called the microglia. It's called the microglia because it's a glial cell that's really small. And of course, we know the saying that things are often small, but they can be very mighty. My view is that these cells are playing mighty roles in the brain. I'd like to show you some data about them. These cells make up only about 10% of the brain, so they're a very small population in the brain. But we're actually interested in what they do. They are unique cells because unlike every other cell type that's in the brain, these cells are not born in the brain. They're actually born outside the brain during development and then migrate and take up residence in the brain. And this schematic shows them being born in the yolk sac, as depicted here in the embryo. Other macrophages that are also born here actually, for a while, go through a stage where they move through the fetal liver, and then they populate the different tissues in the body. These cells, however, do not go through the fetal liver. They go directly from the yolk sac straight into the brain. When they get into the brain, they are actually extremely long-lived in the brain, and remain there for an extended period of time. So these are studies that have been done for a few years ago now in mice, where we can label some of these cells. And as you can see, this is an individual microglia in red that is followed over the course of, in this case, over a year. And that individual cell can still be found in the brain over time. They do not divide much, although in this case, at least in the steady state, they do not divide much. But in this case, you can see one of this cell that was in isolation for a long time eventually divides after about a year or 51 weeks. And then the divided cells, you can see both of them. This has not only been done in mice, but we have carbon dating methods to actually study these cells in humans. And in a similar way, what we see is that these, in humans, they are actually extremely long-lived and can be resident in the brain for decades. In this case, the average median age of each of these microglial cells is about 4 years. So what this means is that anytime you have an injury, or in the context of the studies that are being presented here, anytime there's, for example, a seizure, these microglial cells can detect that and can have some memory for a while they reside in the brain before they are potentially either dying or proliferating or dividing. So they are an important group of cells. We've studied these cells for over 100 years now, and this is what we know that they do, summarized in this schematic. They interact with the vasculature, they regulate myelination, they clear out debris in the brain. But the thing I'd like to focus on is their role as playing an important role in the connectivity of the brain, which is, of course, important in the context of seizures. So, as we all know, and we've heard several times yesterday and today, that seizures are a really common occurrence. They do not discriminate in terms of race or age or gender. They appear everywhere or in every population. However, most of the work even presented at this conference, and rightly so, comes from a studying of seizures relating with the imbalance in neurons. So they are very neurocentric, most of these mechanisms that we've identified and we've targeted. Despite targeting a lot of these mechanisms, we've been insufficient in reducing seizures in a certain number of patients, which suggests that perhaps we should not only think of the brain as a feature of neurons, but as these other cells in the brain may also be playing important, helpful roles in regulating seizures that we can use as a complementary approach to target epilepsy or seizure disorders. And this is where my work where we would like to do some work, where we're trying to target these other cells kind of as a complementary approach to also reduce seizures. So our basic, that's some of the background I wanted to give, and I'd like to present some of the data that we have that we think is very exciting, showing that these cells can be playing roles in different models and paradigms of seizures, and we'd like to eventually translate it into Dravet syndrome as well. So the basic question we ask is, what do these microglia do in the seizure disorder context? And the approach that we take is, at least from the basic science perspective, is a very simple approach. Though to try to inform us on what is going on or what these cells are doing. And the approach is that we ask the question, whatever these cells are doing, when we take them out of the brain, we can ask the question, what happens to the brain or what happens to seizures? If the seizures get worse, we presume that these cells are doing something beneficial. If the seizures get better, we presume that these cells are doing something detrimental. So this is a broad approach that we're starting to use. And there are different ways to get rid of these cells in the brain. We're using an approach that targets a specific receptor, the CSF1R receptor. And this receptor is required for the survival and maintenance of microglia. It's uniquely expressed by the microglia. And there are drugs that have actually been developed, some of which are actually in use in humans, to cleanly eliminate this cell. So we use this approach, in this case in mice, and apply it to the mice. And what you can see here are these microglial cells in green, in both the superficial region of the brain called the cortex, or deeper regions of the brain called the hippocampus, and a specific region of the hippocampus called the dentate gyrus. And what you see is once we feed these mice these drug in the form of their food, as they eat it, rapidly over the course of 7 days, the number of these cells are nicely eliminated in all regions of the brain. I'm just showing you data here for both the cortex and the hippocampus. The reason why this is really cool is that we can eliminate these cells in the brain, and by some mechanism that we do not fully understand, we can leave the brain largely intact. So other cells are not affected, inflammation is not increased, really nothing else is affected that we can identify. And so we can basically ask what happens when we get rid of just this one cell. And this is work that's being done now by a rising third-year graduate student, as well as a former undergrad in our lab who continued in our lab as a technician, having finished undergrad studies. She'll be starting medical school next year. And what Symphony and Jordan did is they took mice that were littermates, and over the course of 7 days, they treated them with the chow that had that drug to eliminate the microglia, or control chow that would preserve the number of microglia in the brain. And then subsequent to that, we started with a chemoconvulsive model of inducing seizures using cinic acid. So after 7 days, half of the mice lack microglia, the other half have microglia, and we expose all those mice to seizures by treating them with cinic acid. And the results that we see are pretty dramatic. I think Andrew did a good job of describing what these seizure scoring is. 0 is normal behaviors, and then as you increase in number, what you see is that there's a greater severity of the seizures. And in this case, we have control-treated mice that have microglia, and what I'll be referring to as PLX-treated mice that lack microglia, as I've shown you. And when we treat the control mice, over the course of an hour of seizures, they peak the seizures around an hour, and then they start to calm down for the seizures for the remaining 3 hours. What we see with the microglia depleted mice is that they start out seizures roughly the same as the wild types, but their seizures continue over the course of an hour, and their seizures are maintained over the course of 4 hours, or at least they recover from the seizure poorly over the course of 4 hours. And this is just summarizing the data from both in both control and the microglia depleted group, showing that they have a much worse seizures. So this implies that these microglia are actually doing something perhaps beneficial, that when we lose them, we see the seizures are much worse. We now ask the question not only in terms of the seizure severity, but the recovery from the seizure in terms of their behaviors. We did some studies. First of all, we measured their weights, the weights of these mice. And as you can see here in the control group, they dip a little bit in weight after the seizures by the first day, but then they recover pretty quickly as they continue to eat and do better, and they restore their weights. The mice that lacked microglia, what we see is that they actually dip very strongly the first few days of recovery from the seizures, and it takes up to about 7 days before they get back to normal. So they have a poorer recovery in terms of their weight. But also when we put them in an open field to see how much the activity is and how much they move around, we also see that those mice that lack microglia actually do very poorly by the third day, though they recover later. I should mention that during the recovery period, we're actually taking away the drug so that the microglia are being recovered. So the microglia come back pretty fast during this period. I'll show you a movie here of these activities at about 3 days. On the left are control mice. Both of these groups have experienced seizures, but these guys on the left have their microglia present, and you see them moving around in the cage kind of like normal. These guys on the right also experience seizures, but as you can see, by 3 days, they're not very happy and they're not moving around, suggesting that these microglia are playing some kind of beneficial role, that once we lose them, we actually have the seizures being much worse. Finally, you might appreciate that we're looking at individual seizures in this case, but what we did was to ask the question, what if they have multiple seizures? can the microglia be playing a role in the second seizure as opposed to just the first seizure? So we did experiments where we treated mice with control chow initially, all the mice, and all the mice also were exposed to chemoconvulsive seizures the first time. So they had equal amount of seizures. And then during another 7 days, we actually treated one group of mice with the drug that eliminates the microglia, and a second group of mice with allowed their microglia to be present. And then we did a second seizure on those guys. And we asked the question, in the second seizure, do we see a difference in the amount of and severity of the seizures? And what we see is that we indeed see that the seizures overall are much worse the second time around, but especially the group that lacked the microglia, this PLX group, their seizures are actually much worse than the control group. So if you have a first seizure, and then you have a second seizure, even the second seizure, microglia are playing some kind of protective role, at least in this chemoconvulsive context. But of course, we all know that chemoconvulsive seizures have their limitations. They don't always recapitulate the seizure phenotype, so we decided to expand these to other seizure models. Now, we don't do electrical seizure modeling, but a colleague of ours over at UVA, Dr. Ed Perez-Reyes, he actually uses several electrical seizure models. And he did this first seizure model with a chronic hippocampal stimulation. I think Cameron described the kindling model here. And he uses this to stimulate the hippocampus with electrodes. And in this case, we actually blinded him to the groups and just told him the different colors of the chow that we fed them. I should tell you that the pink chow is actually the microglial depleted chow. So what you can see here is the time to which they show status epilepticus or severe seizures is much shorter when you treat the mice for 7 days to eliminate the microglia. And the seizure burden, the cumulative seizure burden is much greater, and mortality rate is also much higher. More of the mice die when the microglia are absent in this hippocampal stimulation model. In a second genetic model, combined genetic and electrical stimulation model, Dr. Perez-Reyes actually uses a VGAT-3 model. In this model, mice kindle much faster following stimulation in this specific genetic model. So this is what a seizure looks like during stimulation, and this is what a spontaneous seizure looks like after the stimulation has occurred. So we took some of these mice, exposed them to either control or the PLX chow, and then stimulated them to kindling. What we see here is that the PLX group actually required much fewer stimulations for them to be kindled than the control group. So from these studies, we not only see first in the chemoconvulsive model, but also in the electrical model, we see that a lack of microglia actually makes the seizures much worse. Finally, Symphony actually developed a febrile seizure model. As we all know, febrile seizures are pretty common in kids. So we use a model where she takes roughly about P13 to P15-day-old mice, expose them to a chamber where we can use a heat source to heat the mice, and then they exhibit seizures. In these mice, we actually start to see convulsions, behavioral convulsions, roughly at about 41 degrees or higher. We do not see those convulsions much at lower temperatures. We don't do a lot of electrical measurements, but in a few mice, we're able to measure electrically and show that correlating with these behaviors, we can also see seizure-like activity in both the cortex and the hippocampus in the brain. And finally, we could also document that with this model of increasing temperatures, we actually see increased neuronal activity in the form of C-force expression in both the cortex of mice treated with higher temperatures, as well as the dentate gyrus. So we think our model actually does induce seizures at both the cellular, electrical, and behavioral level. The next question was to ask whether the microglia actually play any role in this context as well. So again, in this case, we treat mice These are neonatal mice, so we can't feed them the chow. We actually have to inject them with the drug. And we can inject them to eliminate the microglia very nicely. This is in control. We see nice microglia in green. In the PLX-treated group, we see that the microglia are eliminated. And then we can measure the seizure activities in these mice. And what we see is that the latency to initiate the seizures is much shorter when we eliminate the microglia. The percent of animals that seize at 41 degrees is almost total when microglia are absent, as opposed to about 50%. And then finally, the mortality rate is significantly greater when we eliminate the microglia. So in all these three models, the chemoconvulsive, the electrical, and now the febrile or hyperthermic, we see that eliminating microglia actually leads to a worsening or exacerbation of the seizures, suggesting that endogenous microglia are actually providing some seizure-limiting roles in this context. We started to try to identify molecularly what those how those seizure-limiting roles are performed. And we decided to focus on this receptor, which is called P2R-Y12. It's a purinergic receptor, present exclusively on the microglia in the brain. So this is RNA-seq transcript data comparing microglia to the rest of the brain. Among all the genes that are expressed by microglia, this P2R-Y12 is highly and selectively expressed by the microglia. We can actually see it here by immunohistochemistry. Microglia are labeled in green here, and we see that the P2R-Y12 is present only on the microglia, co-localizing well. When we perform a chemoconvulsive seizure, we see an upregulation of the P2R-Y12 in both the cortex and the hippocampus compared to in control state. So following a seizure, microglia are able to upregulate this P2R-Y12 receptor. So our question was now, what happens when we get rid of this receptor in mice? Our prediction would be that the seizures would also be worse, just like when we get rid of the whole microglia. And indeed, that's what we see right now in two of the seizure models. In the chemoconvulsive seizure model, we see when we get rid of the P2R-Y12 specifically using genetic approaches, we see that the seizures are much worse. Actually, these mice continue to seize, and most of them end up dying. And that is also shown here in this case. About 75% of the mice eventually die. And then also in the hyperthermic model, we see that the temperature in which the seizures are seen in these mice is much lower. The time to which they begin, in which they begin to convulse is much shorter. And then the percent of mice that actually that actually die during these seizures are actually much greater. So we see that both eliminating microglia generally, as well as specifically this receptor, results in an exacerbation of seizures. So I summarize what we have here, that microglia are brain-resident immune cells. They migrate to the brain early in development. They take up residence in the brain long-term and have the potential to actually have long-term memory of injury phenotypes, including seizures. We show that in 3 different seizure disorders, microglia elimination makes the seizure phenotypes worse. And similarly, specific depletion of the P2R-Y12 receptor also makes the seizure phenotypes worse, at least in 2 models. We're working on the third model to see if that's the case. And yet we conclude that microglia actually perform beneficial roles in acute seizures, and we think that they may be doing this through this P2R-Y12 receptor. This is really cool and unique because this P2R-Y12 receptor is only present in microglia in the brain. So it provides a unique approach for us to target, at least clinically. Now, in terms of our future direction, I've said nothing about Dravet syndrome so far, but we're actually glad to see that recently this study came out just a couple of months ago to show that this P2R-Y12 receptor, which again, like I mentioned, it's only in microglia in the brain, is correlated and associated with epilepsy phenotypes, at least in a Chinese population. This was a small study. They're intending to look at more studies to see polymorphisms that are present on P2R-Y12 could actually be inducing seizures, suggesting that these polymorphisms may lead to dysfunction of the receptor. Dysfunction of the receptor may lead to increased propensity for seizures and seizure phenotypes. We'd like to, of course, extend some of these findings that we have from the 3 different models we have into a more translational model in humans and investigate it with Dravet syndrome. We'd of course like to test both the microglial elimination approach as well as the specific P2Y12 depletion approach in the Dravet syndrome. Now, again, we started to do some of this work. We got some mice from Jennifer Kearney, and unfortunately with the pandemic, we had struggles with getting the line going and keeping the line. But we did have some data, not targeting microglia, but just actually showing that in our system with hyperthermia, we actually see that these mutants, and you've seen this data today from others as well, but we're able to replicate it, that with the SCN1A heterozygotes, we actually see a shortening in time to hyperthermic seizures, as well as the onset of the seizures is actually much earlier. So we think the model is working in our hands. Of course, what we'd like to do is specifically target microglia and P2Y12 here. I should point out, I just added this slide, or this point here, because of some of the things that we learned within the last, actually mostly today. Now, what is interesting to us is that, there's quite a bit of talk that SCN1A is expressed on the axon initial segment region. What is fascinating is that there's clear studies now showing that microglia actually co-localize to the axon initial segment. We don't exactly know what they're doing there, but given that the axon initial segment, it's a region where the action potential is actually initiated, I'm curious to know, even in the DS model, or how microglia are dislocalized in this region. And what is important from this study from several years ago is that at the start of the axon initial segment, you actually see microglia co-localizing increasingly as the mice age into adulthood. So it could be something fascinating to look at. But beyond this, we're also interested in finding out what is the precise mechanism that P2Y12 is eliciting these changes. We're interested in doing some transcriptional profiling of the neurons that lack P2Y12 or that lack microglia to see what changes are present in those neurons to be able to start to pinpoint. But all that I've shown you right now is actually a loss of function of microglia. Either we eliminate microglia or we eliminate P2Y12 and we make the seizures worse. In the clinic, that's not what we want. What we actually want is to improve the phenotype. So we'd like to go the other way, not to lose the microglia or the P2Y12, but actually to enhance their function. And we've generated mice now where we can overexpress the P2Y12. And our prediction is that we would see an amelioration of the seizures or reduction of the seizures, as well as perhaps more clinically relevant to the clinic, using a pharmacological approach. Or maybe Andrew will find a way to help me put our P2Y12 in the capsule so that we can deliver it as well to target it in mice especially. So with that, again, I appreciate everyone in our lab that's put this work together. I was only able to show you work that Symphony and Jordan were doing, and I'll be happy to take any more questions. Thank you.
Do we have any questions for Dr. Eyo?
Yeah, great question. The question is, what's the role of the receptor on the surface of the microglia? So we don't know all what it does. It's a purinergic receptor, so we know that at least it responds to the release of ATP. ATP, of course, is tonically being released by cells. I suspect that it's doing more than just responding to ATP. I suspect that because of the very dramatic phenotype in the seizures, I suspect that perhaps it's responding to ATP and doing other things. It's also really highly expressed in the microglia. I would imagine that it's doing more things than just responding to ATP, but we don't know exactly yet.
So the microglia has been extensively studied in the Alzheimer's field, and the purinergic receptor that you're referring to is a homeostatic microglia population. I was wondering whether you saw any expression of molecules that have to do with disease-associated microglia.
When you say if I saw it when, you mean after seizures?
Yes.
Yeah, that's a good question. We haven't looked at the kind of profile of the microglia after the seizures. What I will say, like you may note, is that P12Y12, you rightly note, is a homeostatic marker. In most pathologies, including Alzheimer's disease, it goes down. What is interesting is that in epilepsy, it actually goes up. Epilepsy is probably the only disease I'm aware of that it actually goes up. So it seems like there's something unique or different going on in epilepsy than in other contexts. What about other genes? I'm not aware of what has been described for other genes in the context of epilepsy.
All right, great. Thank you so much, Dr. Eyo.